Greetings. My name is Roy Scott, and I am one of the USDA National Program leaders who leads some of the plant breeding research that you will hear about during the next few minutes. I'm so deeply sorry that I was not able to deliver this talk to you face to face, but I'm sure you'll understand why. This talk was prepared to give you an overview of ARS plant breeding accomplishments during the last few years to get you started in thinking about what you would want USDA plant breeding to look like in the next few years to come. The ARS budget contains appropriations for research that includes plant breeding. Approximately 150 ARS research projects are involved in some aspect of plant breeding, which is conducted at about 40 U.S. locations by about 200 plant breeders and geneticists working on 60 crop species. Listed here are some of the major crop categories in which ARS has breeding activities, in many cases with strong partnerships with universities and the private sector. ARS plant breeding work occurs under a structured national program that is responsible for implementing the USDA plant breeding priorities. As you know, the purpose of this workshop is to seek input from stakeholders that will be used to design the new USDA plant breeding blueprint that will guide what each of the USDA agencies that are involved in plant breeding will do over the next few years. You also know that ARS plant breeding follows an action plan that is designed from stakeholder input. ARS plant breeding is covered under the main action plan component of crop genetic improvement, which is designed to release new germplasms and varieties. There are two supporting components of the action plan that provide knowledge, research tools, and various enhanced breeding capacities to optimize the breeding. Listed here are the primary breeding goals under the genetic improvement component, which is designed, as I said, to develop superior new varieties and germplasm. In the next few minutes, I will touch on 4 of these goals to give you a brief retrospective view of ARS accomplishments since the last USDA plant breeding roadmap was done. The first example outlines a few accomplishments with releasing drought-tolerant varieties. If you're familiar with soybeans, you probably know that drought tolerance is highly desirable, but is an elusive trait in this crop. However, ARS identified drought tolerance among the USDA soybean germplasm collection and released high-yielding lines that were in turn used by universities and private seed companies to develop high-yielding commercial cultivars that are now in production. In the top photo, you can see a comparison between a new USDA drought-tolerant soybean release and a fast wilting susceptible Czech cultivar. In another example, sorghum is usually grown under dry conditions, so drought tolerant is also highly desirable in this crop. The problem with sorghum is that drought causes both pre and post-flowering stress on the plants. In the past, separate field screening tests were needed to identify plants with resistance to those two stresses, thus lengthening and complicating the process. In response, ARS developed an assay that can be used to identify both pre and post-flowering drought stress, negating the need for long, complicated field screening. The bottom photo is a graphical representation of the test, where high levels of the enzyme durin is shown, along with expression of the stay green trait in the resistant cultivars, in the resistant lines. This process was used to develop high-yielding lines that are now being used by seed companies to develop drought-tolerant hybrids. Here I've described only two examples, but there are many others. For example, with heat tolerance in both lettuce and broccoli, where many heat-tolerant germplasm lines were released by ARS. The next topic I will discuss is ARS breeding for resistance to insect and diseases. Let me give you a couple of examples with insects. A few years ago, the Russian wheat aphid, an invasive pest to the U.S., decimated cereal crop production in Colorado, including feed barley. ARS scientists, using germplasm they identified from the National Plant Germplasm System germplasm collection, released the resistant cultivar Mesa, which brought feed barley production back in western Colorado from a low of 48,000 acres to 78,000 acres in 2015. More recently, a new winter feed cultivar Fortress, resistant to both Russian wheat aphid and green bug, was released. In the second example, the sugarcane aphid migrated to sorghum, creating significant yield losses. ARS plant breeders and entomologists, working together with universities, identified resistance among commercial hybrids and also in the USDA National Plant Germplasm System, and released resistant lines to commercial companies who are rapidly using them to convert the sorghum acreage to resistant hybrids. As with insects, ARS discovered resistance in damaging diseases in most of the crops that we breed. Genes are mapped to precise genomic locations and markers are developed for efficient breeding. One such disease is verticillium wilt in lettuce, in which the pathogen is soil-borne and may persist in the soil for decades. Salinas Valley, California accounts for 50% of the U.S. lettuce production and was suffering catastrophic losses to race 1 of the pathogen. ARS released many resistant lines to commercial companies who are rapidly developing resistant cultivars. You can see in the top right photo the difference between one of the breeding lines and a popular susceptible cultivar. In another example, peanut farmers in the southwest peanut growing regions of the U.S. cannot afford to grow peanuts profitably unless they have Sclerotinia blight resistance. In the last 6 years, ARS released 5 new high oleic peanut cultivars with Sclerotinia blight resistance, which has allowed farmers to participate profitably in a rapidly growing new market opportunity. Yield is probably by far the most important trait in all ARS plant breeding programs. And ARS plant breeders strive to place all other traits they are breeding for into high-yielding backgrounds. I will give you one classic example in which ARS scientists provided breakthrough yield solutions to sorghum producers in recent years. Through mutation breeding approaches, they discovered multi-seeded sorghum mutants with significantly higher yields, mapped the multi-seeded loci to specific chromosomal regions, and developed molecular markers to aid in marker-assisted breeding. They released the lines to commercial companies along with the markers to aid in rapid development of high-yielding sorghum hybrids. The top photo shows a normal hybrid compared to one developed from the USDA germplasm, while the bottom photo shows the genetic changes in number of branches, branch size, and number of seeds that led to the yield increases. Breeding to improve nutritional components of crops has become more prominent in recent years. I will describe 3 examples of ARS accomplishments in breeding plants with superior product quality. In peanuts, there has been a recent new industry need for high oleic peanuts to help with efforts to raise cooking and nutritional quality, along with increased shelf life. In response to this need, ARS peanut breeders released 7 new high oleic peanut cultivars within the last 10 years. In the second example, potato tubers must be stored at cold temperatures, which leads to accumulation of the reducing sugars in a reaction called cold-induced sweetening, shown in the top right photo. During frying, cold-induced sweetened potatoes produce dark, unacceptable products. ARS developed and released several potato germplasms with resistance to cold-induced sweetening, which have been adopted by the industry. You can see a comparison between one of the new releases and a standard potato variety in the middle photo shown here. In the third example, ARS released the first ever true red spinach cultivar in 2019, shown in the bottom photo, which contains higher levels of beta-cyanine and 50% more antioxidant than other spinach cultivars. Now here's a brief look at ARS variety releases in a few crops that we breed, showing several breeders producing more than between 20 and 40 cultivars releases, cultivar and germplasm releases since 2013, encompassing some of the traits that I just discussed with you. In order to accomplish breeding faster and more efficiently, ARS has increased efforts in developing high-throughput genotyping and phenotyping methods to help breeders evaluate the large numbers of progeny required for effective selection. One example is the development of regional genotyping centers, as shown here for small grains. ARS developed 4 small grain genotyping labs, strategically placed and based on wheat market classes where breeders can take advantage of use of advanced breeding technology to develop various types of molecular markers and sequencing. These labs demonstrate effective partnerships among public, private, and international participants. More than 200,000 samples are analyzed at these labs each year, and several new public and private cultivars have been released from this effort. Many ARS breeding programs are adopting high-throughput phenotyping methods to do breeding more efficiently, including development of ground and aerial methods of measuring plant phenotypes. For example, US ARS researchers are developing tractor-based systems designed to be able to screen the large populations needed to conduct meaningful genetic studies under field conditions. These systems monitor physiological responses with much higher temporal resolution and much greater accuracy than through traditional measurements. ARS, in collaboration with external partners, work to provide effective crop genomic and genetic database systems and bioinformatics support for users of the wide variety of crop species that we breed, each demanding individual and specialized tools. Shown here are a few that are strategically placed and most widely used. During the years since the last USDA roadmap, through these databases, ARS has developed new bioinformatic tools for data analysis, developed new tools to manage the high throughput phenotyping and genotyping advances that I described previously, expanded outreach activities, and made database improvements suggested or requested by users. ARS has also expanded the utilization of bioinformatic tools and data by the research community. One of the concerns highlighted during the stakeholder dialogues that led to the last USDA plant breeding roadmap was the need to add personnel and build capacity for high throughput breeding among underserved crop species that we breed. In response, ARS added several new bioinformatic positions and breeding initiatives. In the Southeast area alone, ARS added 3 new bioinformatic positions attached to the same lab as a unit, but strategically placed at different labs within the area for efficiency. You can see here the capacity that one of the new positions adds for breeders of several different crops in analysis and tool development, in genomic sequencing, germplasm characterization, and high throughput phenotyping for underserved crop species. As you well know, the USDA National Plant Germplasm System is the cornerstone of USDA plant breeding. The National Plant Germplasm System continues to conserve plant germplasm, and it enables its users in research and breeding. As you can see, number of accessions remains strong at a high of 596,000, with annual average distribution as high as 286,000 samples during the last 10 years to both domestic and international users. ARS continues to take seriously the need for training and mentoring of the current and future generation of plant breeders. Our scientists continue to find ways of integrating mentoring into their breeding programs to provide opportunities for a diverse range of new plant breeders. In her opening remarks, you heard the enthusiasm that the ARS administrator has for plant breeding. ARS plant breeders have been energized by the innovative programs that she has championed and provided financial support for. For example, Grand Challenge Synergies is an initiative that brings together integrated, synergistic, multidisciplinary teams to solve major agricultural problems, and ARSX provides incentives to encourage high-risk, high-reward research. A third initiative, Breeding Insight, aimed to build teams of specialists to target areas that support plant breeding, especially with underused or underserved crops. So where does ARS go from here? One important goal is to democratize genomics for crop breeding across ARS and to integrate genetic and genomic data to identify functional genetic variation for breeding. We also want to empower breeders with specialized support in bioinformatics, analytics, IT, and high-performance computing so that functional genetic variation can quickly and affordably be combined according to stakeholder needs. Currently, most ARS breeding programs are emerging from Breeding 3.0. Breeding 4.0 is still aspirational for most of ARS crop breeding programs. Breeding 4.x is for discussion and for your input. USDA ARS continues to make strategic investments in bioinformatics, analytics, IT, and other resources for crop, animal, and other organisms across its breeding and research portfolio. I hope you will consider these areas as you provide input for the next USDA plant breeding blueprint. Thank you for listening, and I hope you'll be able to join us at the concluding live interactive session where I'll be happy to answer any questions you may have.